So thanks, Ivan. So yeah, today I'm going to talk about work that I did sort of before coming here. But I thought before I start on that, I'd kind of quickly outline what it is I'm actually going to be doing here at EBD. So as Ivan said, I'm a postdoc in the Ecology, Evolution, and Developmental group, in Ivan's group. And in part, they work on developmental plasticity, which can essentially be defined as sort of the ability of an individual to modify its development in response to environmental conditions. We're probably all familiar with examples of that kind of plasticity. So for example, Daphnia can can develop these quite striking defensive mechanisms in response to increased predation pressures. And another quite nice example are these salamanders, which if environmental conditions aren't right, can perform this neoteny where they become reproductive sort of larval forms, or they stay as larval forms past the reproductive stages. So here in Doñana, we're quite lucky to have a really nice developmental plasticity system right at our doorstep, the western spadefoot toad, Pelobates cultripes. And this frog, sort of the larva of this frog can respond quite drastically to changes in environment. So for example, increased predation pressure, I'm not sure if you can quite see it, leads to this development of really deep tail fins in response to lower predation, in comparison to lower predation pressures. And similarly, when tadpoles are at risk of drying out due to sort of habitat desiccation, they can really drastically increase their developmental rate and sort of shorten their larval period by up to 30 to 40%. So we know a fair amount of what's kind of regulating these changes, especially on sort of the hormonal level. And there's quite a number of studies that have kind of highlighted the importance of thyroid hormone and corticosterone. So this graphic here shows pre-metamorphs and post-metamorphs. And what you see is that when you experimentally drop the water levels, sort of both of these hormone levels surge. There's also been some work at looking at the gene expression, so mostly on Xenopus, which is kind of the go-to model organisms for amphibians. And we know that there's sort of a fairly large number of genes that are directly responsive to different levels of the thyroid hormone. And in Pelobates too, we know that the receptors of thyroid hormone are also upregulated when you experimentally drop water levels. So this is stuff that Ivan and colleagues and his PhD students have been largely working on. And currently we're kind of taking that to the genomics level, looking at transcriptome-wide changes in gene expression under sort of environmental stresses such as reducing water levels. And my project is going to take it to kind of the gene regulation level. So this is an extremely plastic system, so we're expecting there to be a lot of or a complex network of regulatory systems in place. And I'm going to focus on two commonly known epigenetic mechanisms, DNA methylation and nucleosome eviction. So for DNA methylation, we're going to do sort of a preliminary screening of differential methylation profiles using a fairly inexpensive method, sort of a development of amplified fragment length polymorphism. And then we're going to up the ante a little bit, looking at genome-wide changes in nucleosome eviction with a fairly new technique taking advantage of sort of short-read Illumina sequencing. So this is all still really much in the pipeline, so I don't really have anything to talk about here, which is why I'm going to stick to life history in amphibians, but I'm going to talk to you more about the work I was doing before I came here in Switzerland. And that was looking at the evolution of terrestrial breeding in African amphibians. So one of the long-standing interests in biology has been to try and understand all the diversity in life history strategies that are exhibited in organisms. And amphibians make quite an interesting system for studying this because they seem to show the highest diversity of reproductive modes in any vertebrates. And most of that diversity is represented by the anurans, the frogs and toads, which is one of the 3 phyla of extant amphibians. And their plesiomorphic life history cycle, or sort of reproductive cycle, is often referred to as being biphasic. And I'm sure most of you are familiar with that. So that involves an aquatic larval stage. And these larvae hatch from eggs laid in water. And then they undergo this quite drastic change, metamorphosis, to become sort of these ecologically, anatomically, and also physiologically very different terrestrial adults. So inevitably, amphibians tend to be bound to aquatic habitat because of this mode of reproduction. But that's not always the case, and you have sort of differing degrees of terrestrial breeding in amphibians. I guarantee you there's no surfing amphibians. Here we go. Okay, so we have differing degrees of terrestrialization in reproduction in amphibians. So some species, for example, they've maintained the aquatic tadpole stage, but they've lost the aquatic egg-laying stage. So these are frogs like Caramantis that have foam nests, or Alites in Europe. Maybe some of you are familiar with these, where the adults carry the eggs until the larvae hatch into water bodies. And then you have completely terrestrial reproduction. So these are things like direct developing frogs that lay eggs on land, and from these hatch tiny, miniature, perfectly formed adults. And in very rare cases, in anurans at least, you have viviparity, so the giving birth of live young. So they've completely skipped the tadpole stage. If you look at the textbooks, what you often find is that these reproductive modes are kind of listed in an order, sort of from aquatic to terrestrial, and very often sort of the same sort of sequence or linear sequence has been suggested for sort of the evolutionary history of these traits. But more and more that's starting to be challenged, and we know, for example, that these complete terrestrial forms don't necessarily need intermediate semi-terrestrial stepping stones. So they can seemingly evolve from completely aquatic reproductive modes. So when we look at where they occur, these terrestrial modes, we see that they're not sort of evenly spread around the globe, and they seem to be concentrated in the tropics. And when we zoom into the tropics, again, they don't seem to be distributed homogeneously, and there seems to be some structuring. And in the 60s, sort of 2 hypotheses were proposed to explain why you seem to find terrestrial breeding species in some places and not others. So this first hypothesis was proposed by Goin and Goin in 1962, and they seem to stress that steep topography was important for the evolution of terrestrial breeding. So they suggested that in terrestrial montane habitat, where you find sort of more fast-flowing streams and not many standing bodies of waters, you'd either have to evolve tadpoles that are adapted to fast-flowing waters. So here you have a tadpole with these huge suckers to hold on to rocks, or you have sort of tadpoles that evolve limbs very rapidly, again, to prevent them from being washed downstream. And they suggested sort of the alternatives to that would be to just get out of the water completely and evolve and reproduce on land entirely. 2 years later, Poynton suggested that yes, the association to tropical montane habitats was correct, but the interpretation was wrong. And he suggested that what really drives terrestrial breeding is sort of dense undergrowth, sort of moist forests that are often associated to these tropical montane environments that are necessary to prevent the terrestrial eggs from being desiccated, from drying out. So these were 2 hypotheses we wanted to explore further because they were more commentaries than anything. And that kind of was one of the key questions of my PhD. So I wanted to look at whether or not forests and steep topography were important environmental parameters for the evolution of terrestrial breeding. We wanted to develop those ideas a little bit and also look at the life history trait diversity and rates of evolution and things like that. And we wanted to know whether terrestrial breeding could be considered a key innovation. So I'll talk a little bit more about that later, but essentially a trait that promotes higher diversification rates. Both of these hypotheses, the going and Poynton hypotheses, were based on observation of African amphibians, so we restricted our work to African systems, and we took two approaches to this. So the first approach was to look at a specific region, so the Eastern Arc Mountains in Tanzania, and look at how reproductive modes are associated to habitat there. And the second approach was to look at a taxonomic system. And this was the Buffonidae, the true toads, and there we spent a lot more time constructing phylogeny, looking at life history evolution, looking at diversification rates, and also sort of circling back and addressing the question of terrestrial breeding and habitat. So the Eastern Arc Mountains. The Eastern Arc Mountains are a mountain range sort of diagonally dissecting Tanzania in East Africa. They consist of 12 independent mountain blocks, and together with the adjacent coastal lowlands, this area makes up one of the biodiversity hotspots of the world. It's renowned for its species richness, and amphibians are no exception in this case. And we wanted to see if the 180 species of amphibians that occur there are distributed, and their reproductive modes are distributed evenly, or whether we can make some associations with habitat, specifically forest and montane forest habitat. So we scored these amphibians on an ordinal scale for reproductive modes. So we had aquatic eggs and larvae, terrestrial eggs with aquatic larvae, and complete terrestrial development. And we also assigned these species to one of 4 habitat categories. 2 of them were non-forest categories, so we had coastal lowland, non-forest, montane grasslands, and then we had 2 forest categories, coastal lowland forest and montane forest. We took those and we plotted them on our phylogeny, and sort of what was instantly apparent is that especially breeding strategies were highly phylogenetically conserved. So there's really strong phylogenetic signal, especially in breeding biology. But nonetheless, even taking that phylogenetic non-independence into consideration, we found positive association to both coastal lowland forest and montane forest with terrestrial modes of reproduction. And just kind of as an excuse to show you some of the weird and wonderful amphibians we got to work with. This is a Calulina, a direct developer that's almost exclusively found in montane forests. And similarly, these viviparous nectophrenoides are again endemic to the Eastern Arc Mountains. So we knew there was some association to forest, but we had sort of differing degrees of terrestrialization. We had terrestrial eggs and terrestrial larvae. And we also had 2 different types of forest. We had lowland forest and montane forest. So we tried to tease that apart a little more. And base traits is quite a neat program that allows you to test for correlated evolution of 2 binary traits. And essentially how it works is it runs 2 models. It runs a model where your 2 binary traits are allowed to evolve independently on your tree, and then it runs the same model, but where the background state of one trait is dependent on the other. So if you have I can maybe show you an example. So if we have our 2 traits as, in this case, aquatic versus terrestrial eggs and non-forest versus forest, if in the independent model, these 2 traits are allowed to evolve freely, and then in the dependent model, the background state of one of the traits directly influences the transition in the other state. So we did that 4 times for each possible combination. So for example, we have terrestrial eggs versus aquatic eggs, and non-forest to lowland forest, and then terrestrial eggs to aquatic eggs, and vice versa, and non-forest to montane forest, and the same for terrestrial larvae, both with lowland forest and montane forest. What we found was that for terrestrial eggs, the correlated model, so where the two traits are dependent on each other, performed best for both types of forest. But interesting, for terrestrial larvae, we only recovered a significantly better fit for montane forest. So what that means is that, I mean, we do find an association of terrestrial breeding and forest, both montane and coastal lowland forest. But there's also a differentiation between lowland and montane forest, at least for terrestrial larvae. So we find support for both of these hypotheses. So the Eastern Arc Mountains were a really nice system to work with because they host sort of this huge diversity, both in lineages and reproductive modes. But we weren't really that satisfied with the phylogeny we could construct. We were working with very coarse habitat categories, and we never really directly tested this topographic component. So we never really looked at slope. We just used montane versus coastal lowland forest as kind of a proxy. So we thought we could do better, and we decided to look at bufonids in more detail to try and get at those shortcomings. So Bufonidae are the true toads. They're the third largest family of amphibians in the world, with about 600 species. They're neotropical in their origin, but they're cosmopolitan, so you find them pretty much everywhere. Sort of their global success has been attributed to what's been coined the optimal range expansion phenotype, which basically describes the toads we're familiar with, sort of these big stocky things, toxic glands laying a huge number of eggs. And this quite nice study in 2010 showed that shortly after the evolution of this phenotype is when you saw toads getting around everywhere. So in this space of time, toads moved out of the neotropics and colonized all other continents. And here is where they got into Africa. So this previous phylogeny was kind of a global phylogeny, and African species tended to be underrepresented a little bit. So the first thing we had to do was really sort of up the sampling effort, and we managed to collect around 600 tissues, mostly from museums and collaborators. But I also had the chance to go to Africa 3 times and do a bit of my own sampling. So here's us looking like we just robbed a bank in Rwanda, but we didn't, I promise. We had some minor car trouble in Cameroon. And then finally, we decided to give up on research and join the European Coalition of Positive People in Malawi. So with that sampling effort, we managed to get about 70% of all African species represented in our phylogeny. So that's 30% more than any previous published tree. We had representatives of every genera. I put an asterisk there because we failed to get tissues, or at least to amplify any sequence data from this guy. It's a monotypic genus Laurentophrine from the Democratic Republic of Congo, and it's not actually been cited in something like 50 years. So fair enough, I guess. We amplified 5 genes, both mitochondrial and nuclear. We had about 3,500 base pairs to work with. And we took our concatenated alignment with sort of optimal partitioning schemes and substitution models together with fossil calibration points. We threw that into BEAST to generate our working chronogram. So I don't really like showing trees in presentations. I think they're kind of hard to really interpret. But basically what I wanted to show you is that we could confirm that African bufonids are not monophyletic. So they form two clades, this blue clade and this green clade. So this blue clade is distributed pretty much everywhere in Africa. It's got close to 90 described species, compared to this second clade that's slightly younger, really restricted to Central and parts of West Africa, and only has something like 12 species, at least currently described species. So in that first clade, you have your typical optimal range expansion phenotypes, so these big stocky toads, but you also have what I like to call these microtoads, so they're much more smaller and have sort of weird reproductive strategies. And then when you move across to the second clade, that seems to be entirely made up of these very specialized forest toads. So for a number of analytical and practical reasons, most of the work I'll talk about is focused on that first clade. So along with the sequence data, we also collected life history data. We had our reproductive modes, so similar as for the Eastern Rock Mountain project, we had categorical reproductive strategies, but we also gathered quantitative measures of body size, clutch size, and egg size. And again, most of that came from literature, but we also went to some museums and we got master students to dissect gravid females and count tens of thousands of eggs. So what you see is, despite their sort of standard bufo origin, African toads are extremely diverse. So yes, you have these big toads, 15 centimeters is quite large for an amphibian, 15,000 eggs, but they're very small. And then on the other end of the spectrum, you have these tiny little things that lay barely 90 eggs, but compared to their body size, these are absolutely enormous. And when you plot body size and clutch against clutch size, you see the more or less expected trade-off between the more or less expected correlation. So larger females lay larger eggs, but you also see that this pattern is different for different reproductive modes. So terrestrial species have larger clutches, and as you see in other systems, there's also a trade-off. So larger clutches, species with larger clutches tend to have smaller eggs. But what's interesting here is that this pattern was the same for both aquatic and terrestrial species. So beyond that, we wanted to know, what's generating all of this diversity? I mean, where did this all come from? So we wanted to look at the rates of these traits over time. And there's a fairly new software called BAM that allows you to do some of this. So it estimates the rates per trait through time. And what was interesting is that especially for egg size and body size, these rates seem to be very constant. Only really for clutch size do you have kind of a slight increase in rates at the early history of this clade. But when you sort of apply likelihood model fitting approaches, again, we consistently recovered constant rate models as being the best fit for all three of these clades. So there's, seemingly these traits have evolved constantly. There wasn't any burst associated to, for example, moving into a new niche space as one might expect. We also wanted to test whether these rates are different among subclades, and that can be done using a trait disparity analysis. And essentially what that does is it scores the variance or the sort of the spread of variance across subclades, where one is represents trait variance that's highly spread across subclades, and 0 essentially means that your variances are very much divided. So to give you an idea of what that looks like, so if this was our phylogeny and these blue bars represented body sizes, if body sizes sort of evolved randomly as a function of time, what's often referred to as the Brownian motion model, you would expect to see sort of a constant drop-off in subclade disparity. Sort of alternatively, what you can have is that sort of early on you have essentially 3 body forms. You have a clade that's consisted of only small-bodied toads, a clade of medium-bodied toads, and a clade of big-bodied toads. And what that would look like in your disparity plot is sort of a rapid drop-off So strong segregation in trait space early on in the clade. And the extreme opposite of that would be sort of a consistent disparity throughout most of the history. And what that essentially means is that within these three subclades, sort of the variance within that subclade is comparable to the variance of the entire clade. And that's often an indication of convergent evolution. You can get a measure of how much your trait space sort of deviates from the expected Brownian motion model, and that's essentially just the difference in the area between your observed curve and a simulated Brownian motion curve. So this is what our data look like. So again, we have body size, clutch size, and egg size. And all I really want you to focus on here is the dashed line, which is our simulated trait evolution under Brownian motion model, and then the solid line, which is what we actually observed in the data. So what you can see is that these indices, sort of the comparison to the Brownian motion model, are all very close to 0. So there's very little deviation from this constant rate model. So again, we don't find any partitioning of our trait space across subclades. So that's interesting, but we weren't quite ready to give up yet. And we wanted to know whether or not terrestrial breeding can be considered a key innovation. So a key innovation can be defined as a novel phenotype that promotes bursts in lineage diversification. And the reason why we wanted to test this is because if you look at amphibians, you seem to find that the most species-rich families tend to have extremely large numbers of species. And in our phylogeny too, the viviparous nectophrenoides seem to be extremely species-rich. So we wanted to know whether or not terrestrial breeding might have opened up sort of a new dimension in niche space that has allowed these lineages to proliferate. To do that, we, similarly to the Eastern Arc Mountain project, we devised categorical reproductive modes. So here we have 5 reproductive modes, 2 different forms of aquatic reproduction, just open water breeders, the sort of the standard tadpole. Then we have micro water body breeders. So these are species that have tadpoles in tree holes or even in empty snail shells. So that gives you an idea of how small they are. And then there's 3 different forms of terrestrial reproduction. There's direct development, and then there's 2 different forms of viviparity. And that in itself is quite interesting because for a long time it was thought that this matrotrophic viviparity was a direct evolutionary derivative of derivation of lechithotrophic viviparity. But what we could actually show, so here we have the green lechithotrophs and the red matrotroph. What we could actually show is that they've been evolving independently for something like 20 million years. Ancestral trait reconstructions has kind of suggested that their common ancestor was aquatic. So they don't share at least a very similar evolutionary history. What we can do is, with the help of the MUSI algorithm, is we can generate posterior distributions of net diversification that are associated to these traits. So essentially what it returns is these posterior distributions of net diversification traits, net diversification per trait. And if the terrestrial breeders were associated to higher rates of diversification, what you would expect to see is a shift in that curve to the right. Actually, they're all pretty much overlapping. So all of these reproductive modes have similar net diversification rates. So there's very little evidence, despite sort of the unusual richness, there's very little evidence to suggest that, at least in bufonids, terrestrial breeding could be considered a key innovation. So to kind of summarize these last two sections, we do find a high diversity in traits and reproductive modes in bufonids, but they seem to, that there's a trade-off, sort of as most of us are familiar with. So there seems to be an investment either in large clutches or in large eggs. And traits have evolved very constantly over time and also across subclades. So, and finally, terrestrial breeding could not really, or there was no indication to suggest it's a key innovation. So that's quite interesting in itself, because that all seems to suggest that this impressive lineage and life history trait diversity is all a result of accumulation of variance over time. So that's very different to what we're generally exposed to from model systems like Anolis or the Great Lake cichlids. So to take you back to that summary slide of the first part of my talk about the Eastern Arc Mountains, what we could sort of take away from that is that forest as a proxy of humidity and steep slopes as a proxy for lack of suitable water bodies for reproduction were important for terrestrial breeders. So that's something we wanted to test again with bufonids. So we had our phylogeny, we had our life history traits, this time reproductive strategies, and to that we added sort of rigorous environmental data. So what we did was we vetted close to 8,000 locality records, and with those for those we extracted 4 environmental parameters. So those were obtained from remote sensing data. So forest cover, for example, was a measure of the percentage cover per grid cell. Humidity was calculated from precipitation and temperature data. We got a measure of slope from elevation models, and finally we calculated this topographic wetness index, which essentially gives you an idea of whether water accumulates in a place or whether it's likely to run off. So the idea is that it should tell us whether or not you could expect to find standing water bodies in a specific area. So what that looks like is per species we now have a distribution of environmental parameters. So much more information than those 4 habitat categories we had to work with before. So here, for example, you can see the viviparous Nectophrenoides. This is percentage forest cover. So these viviparous species are all up there in very forested areas, and sort of your general explosive breeder is sort of found here in areas with very little forest cover. So we can distill all of that sort of into box plots per reproductive mode. So we have 6 reproductive categories. These are the same 5 as before, but this time we added 1 extra aquatic category. So these are the stream-adapted tadpoles that I mentioned right at the beginning. So they have these big oral discs that allow them to cling onto rocks. And I guess it's kind of getting a bit late in the talk, and there's a lot of colors and a lot to take in, so I'm going to reduce that even further. So we have the blue bars that represent 3 different types of aquatic reproduction, and then the green bars, which are 3 different types of terrestrial reproduction. So that's kind of what I would like you to focus on. So here we have our 4 habitat parameters: forest, humidity, slope, and standing water bodies. And now we can come back to our original hypotheses. So looking at the Poynton hypothesis first. So Poynton predicted that dense forest cover as a proxy for humidity is important for terrestrial breeding. What we actually find is that only one of the terrestrial reproductive modes or species, only one of the reproductive modes is actually associated to high levels of forest cover. And similarly for humidity, again, only one of the terrestrial modes has a sort of an association to high humidity. And also there's not really a clear separation of the aquatic reproductive modes and the terrestrial reproductive modes. One interesting sort of note though, so if we look at the terrestrial modes, these two here are the two different forms of viviparity, and this guy here is a species that lay eggs on land. So that's interesting because Poynton did suggest that the reason why you need this dense undergrowth is to prevent the desiccation of eggs. And that wouldn't really be a problem for viviparous species that sort of carry the young inside the reproductive tract of the female. So that kind of, intuitively, that makes sense. But we still don't really have a separation from, sort of a clear separation from aquatic reproductive modes. So then, if we look at the going hypothesis, so just to reiterate, they suggested that steep slope was important, and what they meant by that is that you don't find suitable standing water bodies for amphibians to breed in. So if we look at slope, we actually do see that all three different modes of reproduction are associated to steep slopes. And when we look at standing water bodies, and if we look at standing water bodies, all three species occur in areas with this low topographic wetness. So essentially meaning there's little collection of surface water in these areas. What's also interesting is that you get a much clearer separation between the terrestrial modes and the aquatic modes, with maybe the exception of these guys. And again, that kind of intuitively makes sense, because this category is that new sixth category, which are the stream-adapted tadpoles. So I mean, and that's again what Goin and Goin were saying. You know, I mean, either you have tadpoles that are adapted to these fast streams, or you get out. So that's quite neat. So what we can take home from that is that again we find that different terrestrial modes occur in different habitats. Forest seems to be less important this time than it was for the Eastern Arc Mountain system we looked at. And slope and lack of standing water bodies seem to be a much more persistent sort of factor associated to terrestrial breeding. And that takes me to the end of my presentation, really. So just to kind of summarize what we could find for bufonids, viviparity evolved twice independently, so that was kind of a neat little nugget that we sort of came across along the way. Terrestrial breeding didn't lead to an increase in diversification in African bufonids. The diversity of life history traits seems to be an accumulation of variance over time, so there's very little to suggest that there was sort of an adaptive radiation or something like that happening. Forest does seem to be important, clearly less so for bufonids, and a much more consistent predictor was steep slope, and that kind of includes topographic wetness, so looking for standing water bodies directly. So, I mean, there's just a huge number of people that were involved in this. I mean, it involved a huge effort in data collection, so I can't really thank everyone here, but these two guys were kind of pivotal. Both of them were my PhD supervisors, Simon Loder and Hendrik Müller. If you want to learn more about our work or just see nice pictures of amphibians, more on my website, and stay tuned for all the cool stuff that's going to happen in Ivan's lab. And thank you for listening.